Hello, welcome to the Assessment, Inventory, and Monitoring AIM Data Use webinar series. During the course of this series, we have been exploring the use of AIM data at various scales. Many of you have previously joined us, but for those of you who are new to Teams Live, you are muted. Please participate by utilizing the chat feature to ask questions. Please feel free to submit your questions at any point. When asking a question, it is helpful if you enter your name. Once questions are submitted, we will publish them for the audience to see. If you upvote another participant's question, it will move up the queue. The presentations are being recorded and BLM users can find them on the AIM SharePoint. The AIM SharePoint and aim.landscapetoolbox.org have links to upcoming presentations. Our external partners are able to watch our live presentations, and we are working on a way to make the recording shareable externally as well. Thanks for joining us today. Now Alex will introduce Allie, our presenter.
Thank you, Alita. Yes, today we have Alexandra Heller, who's a program manager at NMSU down in Las Cruces, New Mexico. And today, Allie Heller will be presenting her master's thesis work on using AIM data to develop ecological site concepts. And yeah, today we also have myself, the AIM terrestrial analyst at the NOC, and Lauren Price, the implementation lead, as well as Alita. If you're watching this recorded or have any follow-up questions, feel free to reach out to any of us on those email addresses listed there. And with that, Allie, you can go ahead and share your screen. Over to you.
All right. Thank you, Alex. And thank you to this group for giving me some time to talk to you all. So as Alex mentioned, I am going to go through the research that I did to complete my thesis with New Mexico State University, which wrapped up this past spring. And this is a project that I am continuing to work on for the next year or so, though the thesis component is finished. So in this presentation, I'll talk a little bit about what ecological sites are and why they're important for land management. I'll introduce the study area and the data set I used, go over the methods and some of the results, and then talk about some of the implications and future directions for this work. So for those of you who are not familiar, ecological site descriptions are a management tool, and they divide a landscape by abiotic properties, such as climate, geology, soils, landform, and hydrology, which vary spatially across the landscape. So in this example, we tend to see shallow rocky soils on the mountains with higher precipitation and more water runoff due to slope. We see deeper soils with more clay accumulation as well as water accumulation in the lowlands, and then variable soils where these hills come in. And the dominant vegetation types also vary across the landscape. So we'll see tree-dominated systems up on the mountains. We'll see a grass shrub matrix on the deeper soils here, and then maybe some pinon-juniper pockets coming in on these hills. So ecological site descriptions are a way of describing and interpreting both the abiotic conditions and plant communities as they vary together spatially. And they include state and transition models, which help to organize information about how plant communities change over time and in response to disturbances, climate, and restoration. And the way that vegetation responds varies by ecological site. So state and transition models are usually represented by box and arrow diagrams, like what you see on the right here, where each large box represents a state. And states represent consequential shifts in ecosystem structure and function. And the arrows between the states that we see describe the mechanisms by which states change. So in this example, we see shrub invasion as a transition T1, between the grassland state and the savanna state. And we see R1, which is fire management and restoration via shrub control, can transition the savanna state back to a grassland state. And we also see T2, grass loss and subsequent erosion, can transition the savanna state to the shrubland state, and that there's not a transition leading away from the shrubland state, which indicates that the shrubland state is highly resilient to change, and there's not a known mechanism that can be implemented by land management to easily transition out of this state. So each ecological site has a unique state and transition model to describe the plant community dynamics. And having this information helps us monitor ecosystems and make educated management decisions. So ecological site descriptions, or ESDs, and state and transition models can be used, for example, to compare the suitability of different ecological sites and states for wildlife habitat. They can be used to predict the effect of disturbances, such as invasive plant establishment after fire. And they can help evaluate whether management objectives are being met within ecological sites and states. So the NRCS is developing ESDs for the United States, though they are not just used by the NRCS, but across land management agencies as well as by agricultural producers. And the intent is to have continuous coverage of this level of information nationally. And as you can see in this map, as of today, there are some areas that have many ecological site descriptions available, which are shown in darker orange, and then some areas in gray and lighter yellow that have no or few ecological site descriptions available. So we need methods of efficiently developing ecological site descriptions where they are currently lacking. And that is where this research comes in. So the goal of this project was to develop a workflow for using existing monitoring data to detect ecological sites in the Rio Grande del Norte National Monument. And secondarily, to create a reproducible workflow that can be used for ecological site development and verification across diverse landscapes where there is existing monitoring data. So I'll introduce you briefly to the study area. The Rio Grande del Norte National Monument is in northern New Mexico. It is one of these areas that is lacking ecological site descriptions, and it is approximately 350,000 acres and managed by the Bureau of Land Management. And I should mention that soils are currently being mapped by the NRCS across this monument with an anticipated completion date of September 2021. So ecological site descriptions have been brought in from the Colorado Plateau to be used in this area, but they are not site specific. So there's a large disconnect in key species and ecological dynamics between what is in the ESDs applied to this area and what we see on the ground here. And some of the main geomorphic features in the monument are the volcanic cones. There are undulating hills and plains, over 40 active playas, and the deep Rio Grande Rift Gorge. And then, of course, all of the more specific component landforms. And the soils in the monument are primarily young alluvial sediments infilled from the adjacent Rocky Mountains. But as you can see, there is a diversity of soil depths and textures. And the main vegetation communities we see are mixed conifer and pinon-juniper forest on the volcanic cones, a shrub grass matrix on the plains, which is dominated by Wyoming big sagebrush in the south, a little bit of black sagebrush in the northwestern part of the monument, and Winterfat in the north. And this is a multi-use landscape. So today, the most important commercial land uses are cattle grazing, wood harvest, and big game hunting. And we know from general land office records that livestock grazing has been ongoing in this area to some degree since the mid-1800s. And the monument also provides critical habitat to a pretty wide diversity of wildlife species. And there are 23 known treatments recorded since 2002 in the monument. So these treatments were to remove woody plant growth and increase herbaceous cover, targeting either trees or shrubs. And there's visual evidence of earlier additional treatments that we don't have records for. So all of these facets of the monument that I've just introduced, the landforms, the soil, the vegetation, land use and management history, all play a part in developing ecological site descriptions. So moving along to the data set, I imagine many of you might be familiar with the Bureau of Land Management's Assessment, Inventory, and Monitoring Program, which comprised the bulk of the data I used, as well as data from the NRCS's Landscape Monitoring Framework Program. And these are just two examples of standardized multi-use monitoring programs that have collectively accumulated data from over 60,000 plot locations nationally. And all of these programs collect plot level plant and soil data using standardized protocols. And importantly, for my purposes, the methods they use are very similar to those described by the National Ecological Site Handbook for medium and high intensity data collection. And I should mention that I was fortunate enough to get to spend 3 years on the AIM field crew in this area. So I entered this project with familiarity both with the study area and with the data set itself. So at each monitoring plot, there is a soil pit described. Line point intercept is collected, which gives us percent foliar cover by plant species, as well as indicators such as litter and bare soil. Vegetation height is collected. Proportion of the plot with large gaps between plant cover. A soil aggregate stability test to look at stability of the soil surface. A full species inventory. And some elements of plot characterization are described, as well as photo points captured for every plot. There are over 200 of these plots in the monument, collected between 2011 and 2018. So this workflow describes how ecological sites are usually developed. So ecological site concepts are formed based on qualitative and observational data, and they're then verified and tested using quantitative field data. And as we all know, field data collection is time and resource intensive. So particularly for large landscapes across which ESDs are developed, it can be difficult to collect as much quantitative data as might be desired. And this image represents the final workflow that I produced, which uses multivariate analysis to detect patterns in existing monitoring data that can then inform ecological site development. So the idea is that this workflow can fit into the larger ecological site development workflow, and where concepts might typically be developed with qualitative data and observation, they can be developed using patterns that have emerged from the analysis of quantitative data. And this is definitely not the first approach to building data-driven ESDs and state and transition models. But what does make this approach unique, along with some variation in the methods I used, is that it is designed to take a large amount of monitoring data that already exists in the landscape, rather than collecting new data for the purpose of developing ecological sites. So I'll go sort of quickly through some of the key methods I used, one of which is ordination, which arranges objects spatially based on similarity. So in a two-dimensional plot space, objects that are more similar are closer together, and objects that are dissimilar are farther apart. And I also used cluster analysis, which groups objects based on their similarity. So ordination and cluster analysis are often combined, and together they can reveal natural groups in a data set and then help to visualize how the groups are related to one another, as well as some of the driving sources of variation among them depending on the methods that you use. So a visual example I like to give is of all of these pieces of candy. We can see that they are similar and different from each other in multiple ways. And here are the same objects having undergone ordination and cluster analysis. So the cluster analysis generated 15 groups based on similarity. And then the ordination arranged the objects and their groups based on one axis on their size and on the other axis on how tasty I personally found them to be. So this is what I wanted to do with the monitoring plots. Of course, each monitoring plot is much more complicated and associated with over 300 potential variables. There's a lot of these plots. So it becomes really important to figure out how to reduce the complexity and noise of this data set to reveal the relationships and patterns that will inform ecological site development. So part of preparing this data was to determine how foliar cover data from the line point intercept method would be used, and grouping plants by characteristics such as their duration, whether they're annuals or perennials, their growth habit and form, so shrubs versus trees versus grasses, and how they reproduce is a way of looking at plant dynamics more mechanistically, and it can reduce potential noise from species misidentifications. So I tried many grouping schemas from very broad functional group classes like tree, shrub, perennial grass, to on the other hand, leaving the species totally ungrouped. And what worked best in the end for me was a schema where species were grouped by genus and then by functional traits within genera. So information about the plant's duration, their photosynthetic pathway, and reproductive mechanism were preserved. And I should mention short-lived native plants, so annuals and biannuals were grouped together. Invasive plants were grouped, and very dominant woody species were left ungrouped as their own species. And this was to capture some hypothesized climatic gradients that we thought these plants in particular might be able to represent. So this schema reduced over 300 species to 97 groups. And I used these plant data combined with percent bare ground and proportion of the plot in large canopy gaps to understand what vegetation communities were present in this monitoring data. And I chose to include these indicators of percent bare ground and large canopy gaps as they can provide insight into the potential for wind and water erosion, as well as how resources are distributed at the plot level. So I was interested in looking not just at these communities as they were represented by plants and species composition, but also by their structure. So to determine the vegetation communities, I used a fuzzy cluster analysis. And you can ignore the axes on this plot, which just represent the coordinate locations of every plot. But you'll see that these colored wedges represent the plots. And the color of the wedge indicates what cluster the plot belongs to. And you can see that wedges of the same color tend to be clustered in space because they are more similar to one another. So we're looking at a representation of 16 vegetation communities identified through fuzzy cluster analysis based on functional group foliar cover, percent bare ground, and proportion of the plot with large canopy gaps. And what I like about fuzzy cluster analysis is it allows plots to belong to multiple clusters. So when plots are represented by multiple wedges, like those circled in red here, that means that they have an affinity for more than one cluster, or that the clustering algorithm could not decide where to stick these plots. So using this particular type of cluster analysis allowed me to flag these plots that had a more ambiguous identity and to create modal concepts for vegetation communities based on the plots that very clearly belonged to those groups. And I revisited these more ambiguous plots later in the analysis, which I'll talk about a bit further on. So again, 16 vegetation communities were identified and summarized by name by the species that were dominant in those communities, as well as the structural attributes when they played an important role in differentiating those groups from one another. And I'll go through a few of these groups just to kind of give you a visual of what they look like. So to develop all of these modal concepts, I calculated the minimum, maximum, mean, and standard deviation for foliar cover of functional groups. And I also looked at the frequency of occurrence from the species richness data. So cluster 1 is driven by big sagebrush and warm season bunch grasses. Cluster 2, also driven by big sagebrush, but with some pinon-juniper cover coming in. And cluster 3, driven by big sagebrush as well, but with an increase in bare soil and the proportion of the plot with large canopy gaps. So these modal concepts can be compared with one another. So in this example, they are all driven by big sagebrush. They all have a warm season Bouteloua bunchgrass component, though it varies across them. And we see that bare soil and the proportion of large gaps vary as well, indicating that there could be some functional differences among these vegetation communities. And just to show you a couple other of the communities that came up, we had a Winterfat warm season bunchgrass dominated community, a closed canopy forest, an invaded grassland community, and a perennial bunchgrass dominated community. So at this point, we have our vegetation communities defined, and now want to understand how they're distributed across the landscape and how they might relate to ecological sites. So I used a technique called BIOENV, which essentially relates your plant community data to your abiotic data. So I was looking at climatic data from the PRISM dataset and topographic variables derived from a 10-meter digital elevation model. And the BIOENV routine looked at the correlation between all of these variables and the plant abundance data. And then it spits out a subset of abiotic variables that are most strongly correlated with the plant data. And I was consistently getting pretty low correlations when I ran this. So I decided to pull in additional abiotic data from the Soil Grids digital soil mapping product. And this did bump the correlation up, particularly the inclusion of the soil pH variable from Soil Grids. And I should note that that we would not expect any set of abiotic variables to completely explain the variability in plant communities, because there's also the temporal component of what has happened at that particular point on the landscape. So land use and management, which were not represented in the BIOENV analysis. So these abiotic variables are a very important piece of the puzzle, but don't explain the whole picture. So I used these variables highlighted in red, which have the strongest correlation with our plant species data, to develop a classification tree. So a classification tree analysis takes a response variable, which in this case was the 16 vegetation communities, and predictor variables, which were those abiotic variables identified on the previous page. And the algorithm can help identify important breaks in the abiotic variables that predict the response variable, or vegetation community in this case. So I built a tree and used the breaks that the classification tree identified to develop these ecological site concepts. And here's a few examples of some of the branches of this classification tree. That there are 13 ecological site concepts in total. I also used box plots to look at the abiotic ranges that each vegetation community occupied, to see whether there were consistently narrow constraints across abiotic variables, which might indicate that a vegetation community belongs to a single ecological site, or whether those ranges were large, indicating that a community is occurring across a wide range in abiotic variables, and that it might be present on multiple ecological sites. And then just to come back to these fuzzy plots, now that I had my modal concepts and ecological site concepts defined, I wanted to see where these more ambiguous plots might fit in. So I reviewed them each individually. I looked at what clusters they had the highest membership value to, how it was different from those modal concepts, and in what ways, whether it occurred on a treatment, what it looked like in photo points, and where on the landscape it occurred, what ecological site concept it matched based on its soil pit and other abiotic variables, and was it different enough from any existing modal concept that it warranted a new class, and then whether it was similar to any other fuzzy plots. So with this, I really wanted to make sure that if there were vegetation communities that were underrepresented by the monitoring data, and that might have only been captured by one or a few plots, that those were not grouped arbitrarily in with another vegetation community. So in looking at all of these fuzzy plots, I identified an additional 5 vegetation communities that were not as strongly represented in the monitoring data. And then I took all of these vegetation communities and the ecological site concepts and put them together to build a bit more of a narrative about the vegetation communities occurring on them. So there were 15 ecological site concepts in total. This is just the first 6 from that final table, which describes their diagnostic abiotic features and the vegetation community dynamics for each, with hypotheses about vegetation responses to management and disturbance when that was possible and applicable. So here's a bit more of a graphical representation of an ecological site concept for the alkaline flats and dry basins and plains, where we see a slope of less than 4.5, and alkaline soils with a high effervescence response in the soil pits. And we know that these vegetation communities all occur on this same ecological site concept. So I calculated new mean values of functional groups and structural indicators based just off of the plots in this ecological site concept. And what we know about this portion of the landscape from General Land Office survey note records from the early 1800s is that this area was described as a gentle rolling prairie with good tall cover of bunch grasses and occasional shrubby growth. So the vegetation community where we see the highest cover of perennial grass might be close to what are referred to as reference states in ecological site descriptions. But this is just to really highlight that we're not necessarily capturing the reference state with this monitoring data set, and we're not necessarily capturing every vegetation community that's possible for any ecological site concept through this analysis. What we are getting is kind of a snapshot of the story that this monitoring data is telling. So here's another ecological site concept that had 3 vegetation communities on plots that were not in vegetation treatments, and another 3 vegetation communities associated with mechanical treatments. And we do see broom snakeweed as a component in all the treated vegetation communities and not in the untreated communities, as well as some other differences. And our General Land Office survey notes told us that in the 1880s, this area had tall sagebrush, tall bunch and grama grasses with scrubby trees occasional. So this may be an area where pinon-juniper is invading into the sagebrush steppe, which is a common dynamic in Wyoming big sagebrush systems. So with more literature review of tree invasion processes in sagebrush steppe, as well as more information from stakeholder groups such as land managers and land users, we can build a more robust narrative of how these states relate to each other and the mechanisms by which they might transition from one to the other. And this ecological site concept occurs on slopes greater than 4.5%, where we have deep soils on small hills and alluvial fans. It shares the same General Land Office notes as the previous slide. So in the 1880s, this area was also dominated by big sagebrush and grasses. But we see on this ecological site concept, our communities are dominated by a cool season rhizomatous grass rather than warm and cool season bunch grasses. And we see that chemical treatments to remove sagebrush with tebuthyron have produced relative to other treatments, low bare soil, and not a lot of large gaps, as well as consistently high cover of perennial grasses. So while we were able to sort of create hypotheses about how these ecological site concepts and their vegetation communities are responding to treatments, there was not quite enough data representing multiple treatments within an ecological site concept to get a consistent response and really understand how these treatments were affecting a single ecological site. So we see some interesting patterns, like pretty consistent responses in chemical treatments, and on the other hand, a pretty varied response to mechanical treatments. And this is across all of our ecological site concepts. And it's tricky also because for the treatments that we have data on, there is no pre-treatment data to compare it to. So this is really just to say that these relationships based on treatment history are preliminary, and we'll need additional data within these treatments to turn these into more robust dynamics. So I defined 3 ecological site concepts for the volcanic cones, 9 ecological site concepts for the plains and the low hills, and 1 ecological site concept for the gorge rim. And inside of the gorge, as well as the playas, those areas were pretty underrepresented in the monitoring data, so they will require additional data collection for further ecological site development. And in order to take these concepts and the results of this research and turn them into ecological site descriptions, the concepts developed through this workflow will need to be verified with additional data collection and through workshops with land managers and stakeholders. So data collection will focus on the vegetation communities that were represented by only a few plots. We'll also look for additional soil and site characteristics that will help match these ecological site concepts to the soil series that the NRCS is currently developing for this area. And we also want to verify the ranges of variability for the ecological site concepts in their abiotic properties and the vegetation communities. And because the monument is an administrative boundary and ecological site concepts extend beyond the ranges of the monument, where there was not monitoring data, we will collect additional data to verify the true geographic extent of these sites. And then workshops will help to ensure that that these site and vegetation community concepts are relevant to land managers, and they'll help with the state and transition model building process. So land managers and other folks who have worked in this landscape for a long time will likely to be able to provide really valuable insight into additional land use histories and dynamics that we were not able to see through this data set alone. So in addition to being used to develop ecological site concepts where they don't currently exist, this workflow could be used in other landscapes to test provisional ecological site concepts and the ranges of variability for the sites and the states that they describe. We can use quantitative values from these modal concepts to build management benchmarks for ecological sites and states. And because many of these monitoring plots are revisited on a 5-year rotation, this workflow could be expanded to track plots and the way that they might be changing in terms of their vegetation communities over time. So though my thesis is complete, I'm continuing work on this project and taking these results that I've talked about and incorporating them into ecological site descriptions for this area. So a large part of what I'm doing right now is taking information that looks like this, trying to turn it into a usable state and transition model. As I described before, with conversations with land managers and some additional data collection. And a part of that is really establishing ecologically meaningful threshold values from these data-driven concepts. I'm also working on a modified particle size class classification for the monitoring soil data. So particle size classes are a broader groups of soil textural and other properties that can provide insight, particularly into how water is interacting in a particular soil. And just kind of another way of looking at these soil pits. And current keys for estimating particle size class published by the NRCS are not compatible with these shallower monitoring soil pits. So I am trying to come up with some new guidelines that might help group these soil pits in a new and meaningful way. And I'll try some of these analyses again with this new classification. I'm also looking at how vegetation height data might be incorporated into fuzzy cluster analysis to come at the vegetation structure of these plots in another way and inform vegetation community groups. And I'll then be applying this same workflow to ecological site verification for the Rio Puerco Bureau of Land Management Field Office. So with that, I would like to thank you for listening, and I'm happy to answer any questions that you might have.
Thank you so much, Allie. That was great. I think we can all agree that ESDs, ecological site descriptions, and state and transition models are incredibly helpful. And so seeing that AIM data can be fed into generating those and improving those is really cool. And that kind of relates to one of the questions that we have here. So we had a question here asking, is there a way that field offices can get similar work done for areas that need new or updated ESDs? Let's do this. Yeah, I love that enthusiasm. So yeah, like, are there barriers to applying this to other areas or other field offices where there's need for ESDs?
I guess the biggest barrier that I might see is just actually making it happen. So I'm really excited to try this workflow in a different landscape and see how it holds up and what might need to be tweaked based on different another landscape dynamic. But I do think that an ecologist and data analyst working alongside land managers who are really familiar with a landscape could go through this workflow or a similar process where they have, maybe a lot of monitoring data and want some clarification and updates to their ecological site descriptions. I think it is very doable. So yeah, I guess maybe the biggest barrier I see is just kind of logistically in putting those teams together and making it happen.
Thanks, Allie. And this is just a reminder, if you do have questions, please click on the little Q&A box and type in your questions for Allie while we have her here. And we have a question here from Scott, who says they agree with hypotheses generated from analyses, but would caution against use of absolute veg composition values or range of values, given confounding nature of the natural and anthropogenic drivers. Have you considered using percentiles as opposed to averages? So for example, 10th percentile for increases or 90th percentile for decreases, or other areas for informing benchmarks that are not confounded by anthropogenic impacts. So hopefully that was clear. Question about effectively creating benchmarks from this process and ways to do that.
Yeah, I think that approaching it through percentiles or more looking at those ranges is could be a great approach. And I guess just another thing to mention about benchmarks is the importance of subsetting a monitoring data set. If you're hoping to both develop benchmarks and then evaluate the landscape based on those benchmarks, so that the monitoring data is not being reused, to both establish the model and then compare against it. So that's something that we've kept in mind for this landscape, is what proportion of the data might we want to leave out for future evaluation. But yeah, I think that there are a lot of ways to look at and summarize these data values other than just taking the mean or some of the other metrics that I've shown here. So yeah, I think that is something that, especially moving forward with actual ecological site development, I definitely am thinking about, and I think it's a great idea to really experiment with all of the ways we might summarize these data.
Great, thanks a lot. And just a follow-up comment there, I think from the first question we had here is that maybe the barrier to to getting this work done in other areas is just the knowledge around the cluster analysis and the stats and the background of applying that technique, which agreed is is a precious resource. So yeah. And again, Scott followed up here with a comment just agreeing with your response that we need to be careful of any kind of circular reasoning when we're coming up with these thresholds or benchmarks. And I think, yeah, you're right that like filtering or being thoughtful about which data we're looking at is incredibly helpful. So last call for questions for Allie. We don't have any queued up right now. And just in case, I do have one more question for you, Allie. I have tons of questions, but I don't want to dominate the Q&A. So you mentioned you looked at the different ecological sites across each of those different treatment areas, and I was wondering, can you use like results from your analyses or this data to help to improve planning of future treatments? If we know specific treatments work well or less well in different ecological sites or different areas or different plant communities. Do you think there's a way that this research could be applied to planning those treatments?
I do. I think that is definitely a hope for these state and transition models that these data will help inform, and just ecological sites more generally across landscapes, is they can be a really important tool to help tailor the treatments and restoration methods that you're using. And yeah, so it's tricky because in this type of analysis, there initially looks like there's a kind of never-ending amount of data, and as you start to subset it by ecological sites and then treatments within ecological sites, the sample size that you're working with dwindles pretty fast. So from these data alone, I'm hesitant to really make any very strong statements about the responses of ecological sites to treatments. I think that with additional data collection to really look at the full extents of these treatments and just get some more snapshots, those could be made much more robust. But I do think that in general, state and transition models are great tools for helping to plan vegetation treatments, and in particular when they are data driven, and you're able to base those decisions based on what has occurred, and then quantify the results. That's even better.
Agreed. Thanks a lot. I appreciate your answer. And we do have one more question here from Patty, and they ask, how did you confirm that the AIM plots did not cross vegetation communities or ecological site boundaries?
That's a really great question. So in looking through the plot photos was a really helpful way to do that. So a lot of those plots that were identified by the fuzzy cluster analysis as being more ambiguous were plots that were running across multiple vegetation communities and potentially multiple ecological sites. So in this analysis, when I saw that, I actually just pulled those plots aside. And I do think that in this area, those instances were flagged by the fuzzy clustering algorithm.
Awesome. Great. Yeah, thanks again, Allie. It was a really informative and really interesting presentation. I I look forward to seeing more of your research. I think that's all the questions we had for you today. Again, if you do have any follow-up questions for Allie or anyone else, feel free to reach out over email. And I think we'll wrap up the presentation there. Next week we have a break from the webinar, so Scott's presentation has been rescheduled. So we will hear from you all again in a couple of weeks. Thanks again, Allie.
Yeah, thank you. And thank you all for listening and your questions.
Absolutely. Take care, everyone.